Well, hello everyone to this final lesson on the topic of nutrients. Now in this last lesson, we're going to be looking at food test. Now we've already talked about the different types of nutrients. We had starch and sugars, fats and proteins. And now we're going to look at what kind of test we can carry out in the laboratory to test for the presence of these nutrients. Now before going further in this video, be sure to take a look and watch the videos of each test on the website first. And the final thing I want to point out is that this topic on the test for nutrients is very, very important. Okay, I think it comes up quite often for practical exams. So you will need to know the steps for each test by heart. You need to know how to describe them. And you also need to know how to describe for every test the observation if the nutrient is present and the observation if the nutrient is absent. Alright, so let's get going. So the first test, the test for starch, should be very familiar to all of you. Right? A lot of you have done it even in primary school, so I will not bore you further. Very simple. All we need to do is add a few drops of iodine solution to a test substance. Okay, usually on a white towel. Okay, but not always. Now, if starch is present, we say that the iodine solution changes from a brown to blue-black color. Now, take note that I always want to mention what the color at first is, and then what the color at the end is. So in this case, the initial color is brown, and the iodine solution changes to blue-black. Right, you need to mention both, otherwise often you will not get the full mark. Okay, if starch is absent, then we say that the iodine solution remains brown. We do not say that nothing happened. A lot of people like to write that nothing happened. But that usually is not a fair observation. Okay, the observation is that the solution remains brown. Great, so moving on to the Benedict's test. Okay, you should have watched the video online. Now the Benedict's test is the test for reducing sugars. And the procedure, you see the star here, meaning that it's very important. Right, what we do is we add an equal volume of Benedict solution to the test solution. Usually, a lot of books write 2 cm cube, so that's 2 cm cube of test solution of the substance you want to test to 2 cm cube of Benedict's solution. Okay, you can use a dropper or something to measure the 2 cm cube. Right, mix well. Okay, mix everything well. Then place the test tube in a boiling water bath for about 2 to 5 minutes. This water bath has to be already boiling. Okay, do not put the test tube inside when it has not yet started boiling. Only after it starts boiling, then place the test tube inside and observe the color change. If reducing sugars are present, you'll note that the blue color solution, okay, the Benedict solution is blue initially, will change to a brick red precipitate. Alright, this word precipitate, you will need to know how to spell it well. If reducing sugars are not present, as absent, then the solution remains blue. Okay, that's quite simple. Now, even though we say that if reducing sugars are present, the blue solution will change to a brick red precipitate, like this one here, there is actually a range of possible results. If there is a moderate amount of reducing sugars, you will actually see an orange-red precipitate being formed. If there's a small amount of reducing sugar, you will sometimes see a green, if not a yellow precipitate being formed. So there's a range of color precipitates that can be produced, and you need to observe and to record down the correct color. Okay, so don't just write brick red without having seen the color properly first. So what are the reducing sugars, right? Which sugars are reducing? Well, what you will see on your textbook in page 55, if you take a look right now, go on, take a look. Right, what you will see is that all of these highlighted in blue are reducing sugars. So only sucrose is not a reducing sugar, right? Not a reducing sugar. And the reason for this lies somewhere in the chemistry of sucrose, which is a bit too complex for us to go deeper in this video, but you can go and look it up yourself. Now, speeding right on to the test for fats, we call this the ethanol emulsion test. Okay, you should have watched the video online. Now, the procedure for the ethanol emulsion test is different depending on whether your test substance is a solution or a solid. So if your test substance is a solution, then you will follow the procedure on this slide. Okay, so imagine, let's say we have a test substance in a test tube. Okay, something like coconut juice. So this is a test solution. Okay, solution form. Okay, what we need to do is we need to add in 2 cm³ of ethanol, mix it well, and then add in 2 cm³ of water. And that's all you need to do if it is a solution form. If the test substance is a solid, what you need to do is you need to first add ethanol to the crushed solid. So this is some crushed solid, maybe crushed nuts. Okay, what you need to do is add 2 cm³ of ethanol, right, and mix it well. Then next, you need to decant. Right, decant means that, okay, you got the solid peanuts crushed here, right, and you will empty out all the ethanol into another test tube containing 2 cm³ of water. So this here is the ethanol being decanted out. Okay, being poured out from this test tube. Okay, it has already dissolved whatever fats is inside the peanut, and it is being poured out into another test tube with 2 cm³ of water. So something like this is decanting up here. Alright, so that's what you need to do if it is a solid substance. So if fats are present, then what you will see is a cloudy white emulsion being formed in the clear solution. But if fats are absent, then the solution will remain clear. Great, let's move on to our last test. The test for proteins, which is called the Biuret test. Right, this Biuret test, you should have already watched the video online. So the Biuret test, sometimes in your textbook you'll see you can use the Biuret solution, but some labs, and actually most labs, our school lab does not have a Biuret solution. So instead, we use sodium hydroxide and copper sulfate. So what do? Well, first we add an equal volume of sodium hydroxide to the test solution. Again, usually we use the value of 2 cm³. Then we mix the sodium hydroxide and test solution well. And finally, we just, using a dropper, add copper sulfate solution drop by drop. And we shake the test tube after every drop and observe for any changes. Now, if it is present, proteins are present, then we will see that the blue color solution changes to a violet color. Right, you should have seen this in the video. But if protein is not present, then the solution will remain blue. Alright, so take some time to digest all that, to really remember and, in a sense, memorize the procedures. Okay, we'll be having a practical coming up soon, where you will do an investigation into a few different unknown foods, and you will apply what you have learned from these videos in the lab. Alright, see you then.